Hello and welcome to the 2020 Brain and Transcranial Photobiomodulation Virtual Summit. I'm your host, Dr. Joe Doduro. My guest is Professor Dr. Paul Chazot. He's Associate Professor of the Department of Bioscience at Durham University, South Road in Durham, the UK. His lab focuses on the biological and biomedical sciences and how the brain functions normally and what goes wrong with the brain in diseases and disorders. He is a fellow of the British Pharmacological Society, got his PhD in 1991, and he was the first to identify the use of photobiomodulation in Alzheimer's disease with an animal model in 2013. I want to thank you for being with us today. Paul?
Hi.
Hi, and I'm going to show everybody where you're from. Hold on one second so that we can get a picture of how far away everybody is. We've got people from all over the globe. And I see that there's a Nottingham. You're up way north. Wait, did I go?
Yeah.
I've never been that far north.
Okay, we're quite close to the Hadrian's Wall for the historians.
Hadrian Wall for the historians, which was a Roman.
Just north of Durham, yeah.
He was the Roman general who subdued the Celts. Correct?
Yep. Yeah, it didn't manage the Scots. They were too unpleasant.
So let me ask you this: you are a member of the Pharmacological Society and the president of the histamine something. I mean, you've done a lot of stuff with a lot of stuff.
True.
Tell me how did you get started in this thing? How did you go from what you were doing for that time to start to look at photobiomodulation and its effects on disease?
Okay. Yeah. So I'm a pharmacologist basically, and my interest is in, as you said, in brain diseases, understanding brain diseases, and I actually a big advocate of the Jim Black approach, so rational drug design. So actually understanding the biology of the system you're trying to change, and also identifying, the appropriate targets that change potentially in the disease you're interested in, and then developing strategies to either upregulate or downregulate, depending on the situation, the biology to give you benefit therapeutically. And you can do this in a lot of different ways. I'm a classic pharmacologist, so we have projects involved in using classic standard synthetic drugs. We also use natural products. I mean, nature is the best chemist, in our opinion. And the third strategy, I guess, is again using nature in a different way, using light. And the different wavelengths of light actually have, as we know for many years, has many useful therapeutic potentials. Has many potential.
I don't know if you're the man for me to talk to about this, but I'm fascinated by those drug companies that take the stereotypic view of the receptors, and then they sit back in the lab and they build the drug that fits perfectly in there like that. Can you do that kind of stuff? Can you
I mean, that's a perfectly reasonable approach. I mean, it's the Jim Black approach, basically. I mean, Jim Black, for those of you who may not know who Jim Black is, would be surprising. I mean, he got the Nobel Prize many years ago now for developing the first beta blocker, and it was because he understood the biology, what was going on in the heart, what you needed to do. So you needed to design a drug that blocks the beta receptor, which was overactive in the heart. And again, very rational approach. And a similar thing he did with treating ulcers, acid secretion in the stomach, identifying the target that did that, the H2 histamine receptor, so developing a selective antagonist to block that acid secretion specific to the target. Again, rational drug design. So yes, it's a perfectly rational approach, but unfortunately, in the last 15, 20 years since that, a lot of pharmaceutical industries went the wrong way, and they went into the combinatorial chemistry route, i.e. needle in the haystack, chucking lots of chemicals at at targets and hoping to pick out one, as a
Oh.
potential drug. So we did go awry in many ways in approaching drug design, and we are coming back to the old ways. Actually, thinking about the biology more and understanding the disease and developing the drug based on understanding the biology and
the biology. You're a neurochemist, and so you're looking at the chemistry of it. I saw, because we're talking a lot about precision medicine, whatever medicine is, and individualized care. And I've seen some of these reports of people, let's say, who have to take an opioid or some kind of drug, and they'll look at their genetic makeup and say, oh, you don't have the receptor for this or enough receptors for this drug, so it's really not going to be useful for you. You can take as much as you want, but it's not going to have the whole effect. That's reality, right?
Up to a point, yeah. I mean, in personalized medicine, there is something to go for it, basically. I mean, you do need to ideally look at individuals, but it's a tough ask, to be honest. I mean, we have got the technology now to understand what individuals' genetics are like, and we can potentially target specific drugs for specific individuals. Yes, I mean, it explains why when drugs were developed, and why you got a wide range of effects, because obviously every individual human is different in the way they respond to drugs. I mean, opioids is probably not a good choice, because opioids is a wonderful drug for treating acute pain, but it's a horrendous drug potentially for chronic treatment. So it's all
one last
there is
question.
rationale there.
With all the parameters that we have with light energy, we have colors, we have intensity, we have frequency, we have power, we have all these different things.
Yeah.
Do you think that we can individualize it that way to affect the cells and the tissues that we want? Is that what you perceive, or is that just my fantasy?
No, that's definitely a rationale. I mean, that's basically, we have to be sure, because not every wavelength is great, particularly in the infrared range. We know, there are certain wavelengths that, are deleterious to the biology. So we do need to think about and use the appropriate wavelength. I mean, if it was a general effect of light, then obviously
go in the sun.
Yeah, exactly. But obviously there's good elements of the light, and there's bad elements of the light, and we have to be very careful which wavelength. That was in fact our first experiment when we started looking at this. To define which are the most appropriate wavelengths to use, because some of the wavelengths we tried initially, had negative effects, serious negative effects.
Well, we're going to see that. Well, what are we going to learn at the end of your presentation? What's somebody going to understand?
Well, you're going to appreciate, yes, a wavelength is very important, and you have to define your wavelength initially. You will find out that actually, especially for some of these complex diseases like brain diseases, which have multiple changes within the disease. So you need a strategy that actually will target multiple elements. So it's not a single target, it's not a magic bullet type of approach that you need for these complex diseases. So that's one thing that actually particular wavelengths can offer to complex diseases, because it actually seems to be, fortuitously, it seems to be able to modulate different targets in different ways, and most of these targets are modulated in a positive fashion. So that's another reason why we work on natural products, because natural products is the same idea. If you're treating complex diseases, you need complex strategies. It's not a single drug for a single target. It's multiple targets. For example, in Alzheimer's disease, you've got multiple changes in the cell. You've got ER stress, oxidative stress, you've got inflammation, you've got all the proteinopathy, but you need more complex strategies to actually give you.
So when you talk about, excuse me, but when you talk about like a natural, it's I think about the cannabinoid systems when they extract this is this THC. Well, it's not the same as the plant that has terpenes and all these other esters and all these other things. Is that what you're referring to? We'll take it in the natural
yes,
form?
Yeah, that's what I mean. Basically within a natural product, it's a complex mixture of chemicals. And if you're fortunate, you've got to pick the right natural product, obviously, that has the key elements within the plant that gives you multiple positive effects. And we've got a number of our plant projects that do that. You know, we've got a number of plant products that actually have multiple positive effects on the system, i.e., you know, help with the ER stress, help with the oxidative stress, help with the inflammation, but proteinopathies simultaneously. Right. And if you actually start to then fractionate those, you start to lose that complex action, which unfortunately it won't make you money if you start fractionating, because that's why drug companies don't go for it, because it's, you know, it's a complex mixture, so you can't make money out of it. They want single molecules, and single molecules are not going to help you when you're treating complex diseases.
Amen. Amen.
Yeah. So you do need nature. Nature fortuitously has given us an opportunity to give us that complex answer to a complex question. And that's, you know, again, why we're interested in light, in particular wavelengths of light, because they seem to be able to modulate different targets. For example, they modify oxidative stress, they modify gene expression. You know, it's sort of almost like a preconditioning type of approach, where you mildly stress the system, induce internal biological changes, which then protect you from future harder hits, if you like. So that's how I look at light. Actually, it's a type of preconditioning strategy. It's mild stress that in the cell that then induces particular proteins, and I will talk about it when I give you my presentation.
We're almost there.
Yeah, some of those proteins that seem to be induced are stress-related proteins that are induced to protect your cells under stressful situations, like in a disease situation. So that's the idea.
Hormesis.
Yeah. So there's some synergy. There's some, you know, there's various things going on simultaneously within the light therapy. I mean, I don't pretend to understand it. You know, we don't understand every aspect of the mechanisms. I mean, that's my big interest in this area, is understanding the mechanisms, and that's why I came into it. To be honest, I was approached now over 15 years ago by my colleague Dr. Dougal, as some of you may know, who approached me to understand how biologically how these phototherapy seems to work. I mean, I was a skeptic back in those days, like I was a skeptic with natural products as well in those days. But now, over the last 15 years, I've grown to actually think that, you know, there's definitely something in it.
Well, that's the key. Every scientist says that. Every interviewer says, when I looked at it and they told me I don't believe it. That's what I love about you guys.
Yeah, until I understand the mechanisms, then I'll start to actually think there's something, you know, there's definitely something in it.
Beautiful. So let's look at your slides.
Okay.
Light at the end of the tunnel. Question mark, Paul?
That's the one.
Really?
Yeah.
Question mark?
Yeah. This is to do with, obviously, can we use light to get to the end of the tunnel of Alzheimer's disease, basically? Can we actually do something about it? It's about time. We've got a growing problem, aging population. It's a massive, massive problem worldwide, and it's going to cost us, and it's in many ways. And yes, potentially, could light be the answer? Could nature give us the answer, actually?
Rage against the dying of the light. Let's see.
Yeah. A bit of culture. Yeah, so a bit of Dylan Thomas. My wife's Welsh, so this is for her. So yeah, a bit of poetry to start us off. And obviously on the slide I've also listed the names of the group, the guys that have been involved in this project over the years. Natalie and Stephanie. Stephanie's actually in the States. She's a postdoc in the States currently. Natalie, they're both postdocing in their respective labs. Gordon was the guy that approached me a number of years ago, over 15 years ago, with the idea of using light, infrared light, specific wavelengths of infrared light, which he had some success therapeutically. And he asked me the question, you know, how do these things work? Is there some biological basis to this? And obviously that's my sort of question. I'm a pharmacologist, so I like to understand how things work before I believe them, potentially. And that's where we kicked it off. And we did the first experiment back in the day, looking at the wavelengths, because again, it's not every wavelength is good in this business. I'm sure colleagues will appreciate that. So our first experiment was to look at actually human lymphocytes and to look at the effects of UV light, again related to aging. And we were looking at the idea which wavelength may be the most beneficial to protect against UV light, because again, not many things can protect against UV light. And it actually turned out, the wavelength we've worked on a lot over the years, 1068 to 1072 nanometers, at the high end was the most effective at protecting against UV light. And it was actually quite impressive. And just to give you a bit of history, again, one of the things we noticed back in that first experiment, that was one of the biological changes we saw, was an increase in inducible NOS, nitric oxide synthase, which, as it turns out, to bring it right up to date, is a potential therapeutic approach for treating viruses. Antiviral approaches, iNOS upregulation and nitric oxide is an antiviral approach that potentially could be useful against our friend COVID-19. And
that was with the ultraviolet light, if I'm not...
That was against ultraviolet, yeah, it was protecting against ultraviolet light. It was looking at aging, actually. It was our first question was, can we get some sort of biological effects against an aging phenomena? So we used a human cell system and an immune cell system to study it initially, and we treated it with UV light and showed 1068, 70 was the best wavelength. I mean, 670 was also effective, but there was actually one or two other wavelengths that weren't effective.
So we're going to look at the slides here, but what you're saying is you used red and infrared light to show a protective effect against ultraviolet light.
That's right, exactly.
And one of the mechanisms that you saw was an induction of nitric oxide synthase.
Yeah, the enzyme. Yeah, so presumably
the whole system go.
Yeah. But it was just it's interesting because actually, like I said, bringing it right up to these days, that is a known effect of this wavelength, and it's a known effect actually as an antiviral approach, and it has been used, defined as a mechanism for doing that. So combination of that sort of approach plus the known effects of this wavelength on inflammation, so pro-inflammation effects as well as later on anti-inflammatory effects, again is a perfect sort of combination to combat the consequences of viral infection. So you've got a treatment, potentially a dual treatment for a viral infection like COVID. It's something we're exploring at the moment to see if that's a go. And there's already evidence for this wavelength to be antiviral from other studies.
We're going to see some of your studies on that and how you're going to implement that as we go forward. I saw that in the slides. Let's go.
Well, possibly, yeah. That's just an aside. Yeah, we're going to focus mostly on the neuro stuff today.
Well, we're going to see the little experiments there, how you did it. Go ahead.
Okay, so I'm going to introduce you to, I guess, this won't take very long. Obviously, Alzheimer's disease is a big problem, and again, all the things I'm going to talk about today, you can extrapolate to other brain diseases because many of the mechanisms underlying, majority, if not all, brain diseases are related. There's a number of crossovers between mechanisms. So some of the effects we see with the light treatment can be transferred into other diseases like ALS, for example, or Parkinson's disease. So just bear that in mind. Again, I don't really need to introduce you to the disease, as most of you know the underlying pathology of the disease, and the disease progression involves a number of different processes. Again, multi-mechanism, as I alluded to in our discussion so far. There's multiple complex things that are going on in these diseases, and they follow a sort of a time course. So you've got inflammation, you've got ER stress, you've got synaptic loss, synaptic neuronal loss, and various proteinopathies like phosphorylated tau and A-beta changes.
I like this slide. I like this slide, Paul, and I'm going to just hover on it for a second, but is it still true or currently true that Alzheimer's is the number one cause of death in the UK?
It's not. Well, it's definitely, yeah, it's getting that way, to be honest. I mean, still cardiovascular is still is still number one, I guess. But yeah, Alzheimer's is over, and it will increase obviously, and it will become number one, I'm sure, in not too distant future. So it is, yes, it's a major problem, massive problem. And if you add that together with Parkinson's disease and other pathologies, vascular dementias and things like that, dementia of Lewy bodies again, which is number two, if you like, in the dementia disease program. It's a massive situation. But the key thing to bear in mind is that a lot of the mechanistic aspects, the underlying mechanisms, are very common between these different pathologies. So if you find a treatment for one, you potentially can then cross over to a number of other pathologies. And we are starting to look at that. I'm focusing on Alzheimer's today, but we've got data looking at Parkinson's disease models and ALS models actually more recently, so motor neuron disease. Again, a lot of the underlying mechanisms are common. There's inflammation, there's synaptic plasticity changes, there's pathological changes in proteins, SOD1 protein superoxide dismutase in the mitochondria in ALS, but there's crossover between amyloid and ALS between those two diseases as well. So there's a lot of commonalities. Okay, so that's, and again, I don't really need to introduce this to the audience. We have, a range of light wavelengths, and again, we've done a lot of work as well as the infrared range. We also work in the visible range as well. We're interested in some of the biological changes in the visible range, particularly related to circadian aspects, and in particular in the phenomena of delirium, which is actually a massive part of Alzheimer's disease and Parkinson's disease. And it is very relevant now with the COVID-19 in ICUs. Delirium is a major aspect of within intensive care, and it's a major aspect within the aging population.
So just 1 second now. Yeah. It's very, they've pointed out a lot that the loss of smell is one of the first things that happens when you get infected. Now, we've found that that's one of the first things that happens in a sign of neurodegeneration.
Yeah.
So could you describe the word delirium? Because that's a British word. And what does that mean for us, and how does it relate to what you're seeing these days? Because I want to have the correct definition. Okay,
so delirium is a mixture of aspects. I mean, what you have in delirium, you have a change in your awareness of your surroundings. So it's like a psychotic type of phenomena where you have visual sort of changes, and it's basically to do with cognition changes, mood changes. So it's a real mixture of things within your your environment. We think in ICU. It's the environmental changes. So, for example, the non-circadian light changes in your environment can trigger different changes in your biochemistry. And one of the key molecules we work on is histamine. And within the day, for example, histamine is actually what wakes you up at the beginning of the day. Okay, so blue light spectrum actually triggers your histaminergic system and actually wakes you up. So and histamine maintains your awareness, wakefulness, and cognitive processing during the day, and then it tails off towards the end of the day and allows you to sleep. Okay, so delirium we think is actually because the histamine levels are changing all over the place, or going all over the place during the day in an environment like intensive care, for example. And also, if you're isolated, as we are now, in our homes, away from the light, and away from the circadian stimulation, then the chances of inducing a delirium state, where you lose that sort of understanding of your environment, you get delusional, you get you have changes in your awareness of yourself and all around you. So it's a very serious problem, and it's also, it affects your cognitive processing and memory processing as well. So there's a real mixture of things.
That's why people are taking like the antihistamines to fall asleep, huh? Boom.
Yeah, that's at night. Yeah. So yeah, antihistamines. antihistamines, yeah, are involved in if they get in the brain. You know, the classic first generation antihistamines get in the brain. The second generation don't actually. So they don't cause a problem within the brain. But actually, interesting you say that, because if you block specific histamine receptors in the brain, you can potentially cause or induce a more delirious state during the day. So they, patients or clinicians try to avoid using H2 and H1 antagonists that get in the brain to stop you, going into a delirious state. And anybody's interested, I've written a review on this, this idea of histamine being a key factor that we need to manipulate during this situation we're in, actually, this sort of isolation situation.
Pandemic, we can say it. Pandemic.
Yeah. So, it's...
And the clinicians who have to be in the hospital.
Yeah, they're affected as well.
Influenced very seriously, not only by the toxic environment, if you will, or by the viral environment, but also the
stress. Yeah, it's massive,
everything like that. So go ahead. I like it.
Yeah. So we're interested actually in the staff and the patients and the carers. All three of them really are going to be affected by this present situation. So we need to think about that in design of intensive care units in terms of the light and the environment. And so that's one of our projects we're interested in terms of the visual end of the spectrum. But if we're going to focus on the infrared range, so this is basically how I look at it. As I've already mentioned, it's a preconditioning process. So photobiomodulation preconditioning of the brain is what we think is what we're doing by treating individuals with different wavelengths. So we're stimulating a mild stress response within your tissue, and that causes changes, genetic changes, biological changes that will potentially protect or improve the quality or the viability of the cells, be they neurons, be they glia, be they cardiac cells. Any cell in your body potentially can be protected in this way. So the two wavelengths that we favour, many of our colleagues have done a lot of work in the 670 range. There's clear evidence, clear good evidence base for 670 nanometer range being therapeutically useful. We focus most of our work towards the other end, towards the 1070 region. Now I put in 850 nanometers range. I go back to our first experiment. When we used 850 in our lymphocytes, we got actually a very negative effect. So we've avoided 850 since then. That's over 15 years. I know colleagues have found some positive effects of that wavelength and around that wavelength, and even in humans, there's trials with that wavelength and showing beneficial effects. But we've steered away from it, based on that first experiment, to be honest.
So can I just interject, because your group has really focused on the far infrared, if you will. Absolutely. Not so many people at that time and over this last period have kind of gone into the
true.
Higher nanometers. So this is kind of the reason why you shunned away from the 800 range.
Yeah. It may not be a good rationale, because it was really only based on one experience.
I understand. That's kind of what I was going at, because everybody else sort of got away from the higher ones because they were afraid of the thermal and all this other stuff. So
Sure.
I see how you've gone your path.
Yeah,
now,
that's good.
We're going to talk about all these are very interesting. So RNA and DNA synthesis seems to be a big deal these days. People talking about that, and everybody's talking about proteins, but you call them chaperone proteins. So that's a new term for me. So where are you going to take us in this little...
Okay, so I've focused on one part of our story, basically. Chaperone proteins are these sort of proteins that are induced during this mild stress response, as I said. These proteins are there as protection proteins, if you like, and they protect in various different ways against oxidative stress, ER stress, proteinopathies. They have different roles, which we'll go through later on. And the reason I'm focusing on this is we found in our Alzheimer's disease animal model, when we did those first studies, with our wavelength, we got quite profound effects on these chaperone proteins in terms of inducing specific members of these chaperone proteins, which when put together went along with the ideas, the other effects we saw, i.e. the effects on mitochondria, the effects on proteinopathy. So it all made a nice little story that these chaperone proteins maybe were the reason why we saw the other effects, the consequential effects, and ultimately effects on behavior. So yeah, I'm focusing today on chaperone proteins, but obviously we've seen other changes as well from other studies, but chaperone proteins is what I'm going to focus on today because I haven't really presented this as a presentation before. So I put this data together. Some of this data, a lot of this data is not published, so some of these other aspects. So that's why I'm presenting them today.
I'm glad you had a lot of time to think about this because it's going to be, your thinking is going to be helpful. Go ahead.
Yeah, it's been beneficial. I mean, there's one positive thing that's come out of this COVID-19 is actually it's given us time to think about things, design experiments and to think of, yeah, when you haven't got children, of course. Obviously, we're lucky my children are older, so I can focus on thinking about experiments. Okay, so yeah, so again, I'm just talking about things that people know. Obviously, the wavelength was originally selected. It wasn't selected by me. Obviously, it was Gordon with his initial clinical uses of this particular wavelength. I then started looking at the wavelength and why he had selected it and why he was looking at it in his studies, in his early studies. Obviously, it does penetrate quite deeply into tissue and into, which is quite beneficial. So it gets to targets, far-reaching targets potentially in if you're thinking human into the brain. Something I actually, again, I'm telling you my secrets. Basically, I was a very naive scientist in this field when I first got into it. I thought, okay, you've got a light wavelength, and in how surely if you've got a skull, that's gonna stop the penetration of the light into the brain. Then I started talking to guys that know what they're talking about, i.e. guys in physics, and they explained to me how the skull actually is almost like a focusing tool, an optical channel for the light, and it actually allows penetration of the light deeper into the brain, if you're thinking about the brain. So if you take the skull away, and there's been experiments showing this, if you take the skull away, you actually get less penetration of the light into the brain. So again, it sort of started to make sense that this might be a viable strategy in humans. So yes, so the wavelength, yeah, it allows good penetration is one good factor about it if you want to get into the brain and further into the brain, into some of the key centers within the central part of the brain, because then this wavelength, is a potentially suitable one to look at. So this slide just really talks about some of our methods, if you like. And so we use a range of preclinical methods, model systems. We use cells, neurons, glia and culture. And we use various in vivo models, simple ones like C. elegans, which are very elegant models for aging. They have limitations, obviously, in terms of their nervous system. So we then are starting to use more recently going into flies, Drosophila flies, which have a more sort of developed neuronal system, so more relevant to transposing it into mice and then into humans. So we've got various animal models we use within our work. So we can look at, particularly in the C. elegans and Drosophila models, we can look at things like aging and lifespan and healthier lifespan experiments to see, further effects of the light. So we can do things in the Drosophila, we can do things in the mice related to behavior that's relevant to humans. So we can look at motor, memory, and social interactions within these species, particular the mice and even the flies. Now we're developing sort of all-in-one tests to look at different behavioral paradigms. Also in the flies, we can look at disease models, obviously very off the shelf almost. You can get genetic versions, familial genetic versions of diseases, and we've worked with some of those related to Parkinson's and Alzheimer's. So those are the models. We started very early on, obviously, not just us, other studies in the 617 meter range, looking at mitochondria. And it's an obviously biological target for something like light of different wavelengths. And we, like others, have found that our wavelength does have some influence on particular parts of the mitochondrial function, particularly complex 3 and 4. We seem to have some increase in activity, particularly in state 3, which is relevant in terms of ATP production. It's relevant to the disease. For example, Alzheimer's disease, you get significant changes in complex 3 of mitochondria, significant reductions in the temporal lobe, for example, in Alzheimer's patients. So it's very relevant to the actual disease in terms of mitochondrial function. So we've done some studies in purified mitochondria from mice to look at these. This is unpublished data, actually, to look at these initial effects, because as with many infrared wavelengths, the mitochondria seems to be one of the main focal points, if you like, for the mechanism. Following on from that, again, this is unpublished data, actually, where we've looked at the wavelength in cell culture. So we can use primary neurons in culture. We've also done some work in glial cells as well, again, which I won't show today, but we know it does have some beneficial effects on glial pathology, physiology as well. In this study, we were looking at glutamate-induced toxicity and shown protective effects with the light treatments. Again, the key thing we found very early on when we're doing these studies, that you need to do multiple exposures to the light. It's not just a single exposure paradigm. For most of these preclinical studies, to get optimum effects, you need to do multiple, for example, 4 or 5 short-term treatments to see beneficial effects. So again, that presumably is something to do with the mechanistic aspects of the treatment. So we got neuroprotection in those studies. We also did some studies, again, pretty artificial studies, if you like, but you can do look at amyloid-induced neuronal death, and again, we saw some clear protection from that process. You can make beta amyloid, you take it off the shelf, and you can actually generate oligomers and aggregates of the amyloid, and then apply those to the neurons. Again, it's pretty artificial, but actually, when we looked at the EM of those neurons, you could actually see the amyloid inside the cell, which is obviously the most relevant to the disease. It's not, now we know, it's not the plaques that are relevant, it's actually the oligomers, the soluble oligomers that get in the cells that are relevant. So this model actually did mimic that. We did see, presumed death due to the oligomeric structures. So we got neuroprotection. Now I'm going to focus on effects mostly in vivo. So again, this is in mice. Okay, so we're looking at different changes with the treatment. So again, it's using our wavelength 1068 to 1072 nanometers, and in these slides we're just focusing on synaptic plasticity changes. So I'm just highlighting AMPA receptors, which are glutamate receptors involved in synaptic plasticity, physiology of synaptic plasticity, CREB induction, and BDNF. Again, just 2 examples of molecules that are modulated by AMPA receptor activation and synaptic plasticity. And we looked at those in...
Oh, wait a minute, go back, please.
Sorry.
Now this is a...
So I'm just focusing on this.
I got you, but this is a Chinese menu here for me, and it's in Chinese, so help me out here. We got some... Okay,
it is a complex slide, so...
let me just ask you one question now. Let me walk through the thing. If I'm not mistaken, you can use your little mouse though, but below the AMPA receptors, that looks like complex 4 right there, correct?
Yeah, no, sorry. So what you can see below that is a range of changes. These are all signaling molecules we're looking at here. So this is phosphorylated receptors. So don't worry about the complexity of the slide. What I'm trying to just focus is the effects. AMPA receptors are fundamentally important in inducing synaptic plasticity.
I see that
Glutamate
long-term
receptors, yeah.
potentiation and neurogenesis by BDNF. That part I see.
Yeah, that's the neurogenesis process, but also there's effects, on other signaling molecules. mTOR is also another molecule that's involved in synaptic plasticity. So yeah, so it's AMPA receptors control synaptic plasticity, but also they control neurogenesis and long-term plasticity through BDNF.
Good one. Now, where is that located? Where is that receptor located? The AMPA receptor.
Okay, so this is on the postsynaptic side of the synapse. Okay, so you're releasing glutamate from the outside onto here. So as well as AMPA receptors, what's not on this slide is there's NMDA receptors as well. So there's two types of glutamate receptor here. And the AMPA receptors is the one that changes in plasticity. What you get is an increase in number of AMPA receptors here. So it makes it a bigger signal, if you like. So it's long-term potentiation for colleagues. So you get increases in AMPA receptor function and increase in AMPA receptor numbers in synaptic plasticity, underlies the plasticity process short term, and then you get effects long term on gene expression.
Very good.
So protein changes, neurogenesis, synaptic, generating more synapses and more spines. So it's all part of the long term plasticity process.
So answer me this: the GLUT-R2 and the GLUT-R1 represented by the
there's some
in the green are simply, that thing is just simply showing us a receptor. Nothing.
Yeah. Nothing more than that. Yeah, these are subunits of the receptor. So GLUT-R1 and GLUT-R2 are subunits. AMPA receptors have this key subunit, the GLUT-R2, which actually stops it being a calcium channel. So if you didn't have GLUT-R2 or you had mutations in the GLUT-R2, you would be dead, basically. The neurons would be, So this controls calcium entering the cell.
Prevents
so calcium only allow sodium into the cell. So they depolarize the cell.
Boom.
So once when you see depolarize the cell, then you can open the NMDA receptor, which sits next to it, and then you generate the synaptic process, the long-term potentiation.
That's the plasticity effect that you're seeing. It's going to happen right there. That's how plasticity happens. And so
that's how we believe plasticity happens and leads to memory process, generating memories.
Cool.
Okay, is that enough? Yeah, sorry, it's a very complex slide.
Oh, you did great. You did great.
The main thing is to just highlight these three key molecules that I'm going to show you are affected by the light therapy or the light treatment. Okay, this could be a little bit more complicated. Anyway, this is the effect on the AMPA receptors. Okay, this is some very old data, which again we haven't published, but what we're looking at here, if you look very carefully, these are slices from the brains of the mice, and they're labeled with tritiated AMPA. So AMPA is the ligand that's binding to the AMPA receptors. Okay. But it only binds really in this situation to the high affinity AMPA binding sites. Okay, and what you see, what we measure there, and this is hippocampus. This is the hippocampus, and this is the cortex. If you can see that, see my arrow there. So it does label, key structures where AMPA receptors are, which undergo synaptic plasticity, etc., etc. So what we're looking at is labeling. What the light does is actually knocks that labeling down, so you lose the high affinity AMPA sites. Okay.
Right.
Those high affinity AMPA sites we believe are intracellular AMPA receptors, i.e. non-functional AMPA receptors. Okay. So it's only the low affinity AMPA receptors that are actually functional, that sit in the synapse. So what the interpretation of this is that you're losing intracellular receptors, and the presumption is they're going into the synapse to become synaptically functional AMPA receptors.
Ooh, and that's a good thing. That's a good thing.
That's what I mean. It's a good thing. Yeah, it's a bit of a complicated explanation, but that's basically what we think is happening here.
A little bit counterintuitive. So what's the XO? What's the EXO in your little diagram over there?
So it's export or import, basically. So AMPA receptors either sit inside the cell, or they go to the synapse.
They're
the high affinity are the ones that are sitting inside that are non-functional.
Gotcha.
So you're losing those, and presumably you're getting them imported into the synapse. That's the assumption. We haven't never proven that, but that needs to be done. But that's the interpretation of this study. Yeah, so it clearly knocks that high affinity down all over so the assumption is that more receptors are now at the synapse to undergo synaptic plasticity. Okay,
got it.
It's not proven, but that's the interpretation of that data. And then we see clear. These are changes of BDNF expression over time. So this is a CD1 mouse, a male CD1 mouse we found in our studies back in the day, and really has been understudied, is a very good model for premature aging. Because these CD1 mice, particularly the males, have very poor learning performance, even at about 7, 8, 9 months, which is very unusual for a mouse. So they have premature aging. And part of the reason they have premature aging is because they lose BDNF very rapidly here. And what we see when we treat these mice at 12 months or so with the IR, you get an upregulation. So you get a return of the 12-month BDNF back more up to the earlier time point. So you get increase in BDNF expression, you get increase in CREB, again this gene expression that's very useful. That's both in the CD1 mice, which clear upregulation here, but also in these AD mice, Alzheimer's disease mice, that I'm going to talk about a bit later as well. Okay, so in both these sort of aging mice, premature aging mice or the Alzheimer mice, you get this upregulation of CREB as well.
So one second here.
Yeah.
For people who aren't familiar with these, that's the blot, right? That's the blot protein.
That's a blot, yeah. So we're quantifying. These are 4 replicates, 4 replicate animals. Okay, and this is beta-actin to standardize the protein. Because obviously you're loading, you have to standardize the loading, so it's a relative change relative to beta-actin, which doesn't change.
Got it.
Okay, so it's a loading control. Okay. So this is the quantitative data here showing the upregulation of the protein you're interested in. So that's CREB1, before that was BDNF. So that's BDNF changes. That's the beta-actin, which doesn't change, to standardize it.
So we can see that the darkening and the thickening of those lines means there's more proteins in that section. That means that's a good thing. I got you.
Okay, so it's upregulation of BDNF, upregulation of CREB, phospho-CREB as well, actually, which we're not showing. Again, if you go back to that original diagram, so you get changes, presumed changes in increased AMPA receptors, changes in CREB DNA production of BDNF, upregulation of BDNF, which is, again, neurotrophic factors, very good for neuroplasticity, very good for synaptic plasticity generally.
That's the brain fountain of youth.
Indeed, yeah. And things like ampakines and things like, what else? Even, this new ketamine idea with depression increases BDNF expression. So it's one of the mechanisms they think why ketamine is potentially a useful for plasticity, useful for depression type phenomena.
Beautiful.
Just to compare it between the two, yeah. So mTOR and BDNF upregulation is related to the ketamine's effect in depression. I'm still a little bit skeptical about that because it's still quite difficult to explain how ketamine does it specifically, but that's for another day. Anyway, okay. So those are the three effects first. Now I'm gonna ready. Now I'm going to talk about, we talked about chaperone proteins. So I'm now going to focus on chaperone proteins. So this slide is, again, it's just depicting some of the key chaperone proteins I'm going to talk about, just to give you an idea of what they look like. But we'll focus more later in later slides on their functions, okay, specific functions. So, there's been a long history of the relevance of heat shock proteins in particular. These are the chaperone proteins we're going to talk about in degenerative diseases like Alzheimer's disease. So it's already known that these chaperone proteins are modified in these diseases. So it's perfectly relevant, perfectly rational to have a look at them in
when it does this. So let me understand this.
Yeah.
You want to make that effects of 1068 nanometers, so that's clear.
Sorry, yeah.
Upon the chaperone protein, and this specific chaperone protein is the heat shock protein, is right? Because there's probably more of them, correct?
Yeah, of course, yeah, okay. Absolutely. Yeah, sorry, these are heat shock proteins predominantly that we're talking about. And we're going to focus initially on these older CD1 mice, these male CD1 mice which are premature aging, as I said. They have learning deficits. We're going to touch on that in one of the subsequent slides, the learning and memory effects in these mice. So we're looking at the molecular changes in the same mice. We're going to talk about the behavior later. So these are 12-month mice which have serious learning deficits, basically, at this age. And we're looking at various heat shock proteins, chaperone proteins. So initially, we'll just have a look at heat shock protein 40 and 70. These are key ones that are involved in various processes. They're involved in mitochondrial function. They're involved in proteinopathy, protein processing, protein folding, etc., etc. And they're very important to maintain a correct protein folding and processing. They're very important in mitochondrial viability and mitochondrial function. But they are lost in aging, and they're lost to a certain extent in Alzheimer's disease. Okay, so in these two cases, again, I'm just giving you a snapshot of some of the results. So we see upregulation as with heat shock protein 40 and 70. Here, again, these are similar types of experiments. Again, replicates of different animals with the standardized protein. So we get upregulation of heat shock protein 40 and 70. And there's a little bit of immunohistochemistry to go along with it. Again, heat shock protein 40. This is just focusing on one part of the brain, the dentate gyrus, which is part of the hippocampal formation. And you can see, hopefully you can see upregulation of the protein. So this is staining of heat shock protein. It's sort of old-fashioned immunohistochemistry. You don't see this very often these days. It's more, immunofluorescence these days. But the immunohistochemistry, you see upregulation of heat shock protein 40, and I've just shown you a little bit of cortex as for something else to look at. So prefrontal cortex, again, very important in learning and memory processing. Again, this is a heat shock protein 105, which is actually related to heat shock protein 70, so they have similar functions. Again, you see an increase in signal, this brown staining in lamina of the cortex in these mice. These are just snapshots. Obviously, we see changes in other parts of the brain as well. It just gives you an idea. Again, this is a time course again in the CD1 mice. This is heat shock protein 105, which I've just mentioned. Here we see downregulation in aging. It's not as stark as some of the other heat shock proteins, but it's a reduction at 12 months where you've got premature learning deficits. Again, you get upregulation with the infrared treatment. Here by blotting, and here by immunohistochemistry. Again, you can see clear labeling here where it's fairly faint in the sham treated animal as an example. So you get changes in various heat shock proteins with the treatment. So these are multiple, just to emphasize, I haven't really talked about the treatment. So the treatments were done over a 3-month period with these mice. So maybe, let's have a think, it's about 5 or 6 exposures a day every 3 or 4 days, that sort of type of regime. So multiple exposures over the time period. Kept my student busy for 3 months. And then we process the tissue and do the learning and memory performance with the final animals. So these are changes we're seeing. So again, this is heat shock protein 27, a different one, maybe not quite so clear here, but there's an upregulation in the dentate gyrus. This is changes in heat shock protein over time. Again, quite a significant change in the chaperone protein. This is at 12 months, quite a massive change in the protein. You can see that in the plot. Again, you get upregulation of this particular heat shock protein as well. What I need to emphasize, you don't get upregulation in every heat shock. So it's quite a selective pool of chaperone proteins that are upregulated. Okay, so there's a degree of selectivity in its action. And all the ones I'm focusing on are ones that can be tied into functional consequences, if you like.
Mm-hmm.
All right, so there's a range of, so I'm just showing you the CD1 data first. So these are male CD1 data with premature memory performance. So this was the study we published back in 2008, actually it's quite a few years ago now, where we showed the treatment, 3-month treatment, 3-month animals, which are sort of young, healthy animals that are performing well on the learning and memory task. This is a type of our version of the radial maze. It's a unique test, actually. It's an all-in-one test that measures motor activity, anxiety, and memory in the same test system, if anybody's interested. And this testing system saves on animals, and it also allows you to measure all the different behaviors in the same single set of animals. So within that test, we showed that the treatment allowed the animal to perform as good as a 3-month animal. So a 12-month animal that has serious memory deficits. I'm not showing you the data in this.
Right.
Anybody interested can obviously look at the paper. You can see that it reverses the memory deficits to a 3-month healthy, well-performing mouse with the treatment, a 3-month treatment. Oh, actually, no, this was a 2-week treatment. This was a relatively acute treatment with the light. The important thing also is that there was no negative effects on other paradigms like anxiety or motor behavior. So it was a selective action on the working memory, which goes along with this idea of changes in heat shock proteins and goes along with the idea of the changes in BDNF and CREB that I talked about. Now we're going to look at exactly the same thing in some Alzheimer's disease mice. So these are double mutant Alzheimer's disease mice. These are females which have a more serious sort of memory deficits than the male counterparts in this particular model. We're looking at 12-month animals, but we also looked at them over time as well. So this is treatment again with the 1068. We're looking at focusing on heat shock proteins again, but I'm going to introduce you to another heat shock protein, if you like, alpha B crystallin, which is a protein which we looked at as well within these studies. And it's known that alpha B crystallin its expression is changed in Alzheimer's disease neurons, and it modifies beta amyloid aggregation. So it has initially beneficial effects, but then as it increases in amount, it starts to induce further aggregation of the beta amyloid. So there's a positive effect of alpha B crystallin initially, and then it gets swamped, and it actually induces a more severe form of amyloid deposition.
So
okay,
let me just walk me through this a little bit. So we have the A beta crystalline down at the bottom looking like a pretzel, right?
Yeah, that's right. Yeah.
So what's this target protein monomer? It can be bundled up, or it can be so then it's going to be target is intermediate, then it's prefibrillar, then it becomes the target intermediate again. So it all gloms together and then it sticks onto the amyloid fibril?
Initially, like another chaperone proteins, the idea is that it's trying to stop the protein from aggregating, i.e. it's basically trying to maintain its normal structure. So that's its primary role initially, but in Alzheimer's disease it seems to get overexpressed, and you'll see that in the mouse. And the idea is that once it becomes overexpressed, it starts to then cause the oligomers to then further aggregate and form the fibrils in more of a severe structure. So it's believed to be potentially good initially, but if it's then not controlled, it starts to cause negative effects and causes protein aggregation.
Thank you.
The target protein we're talking about here is amyloid, the amyloid structure. So they do interact. It does act as a chaperone protein. And part of the role of chaperone proteins and heat shock proteins is to process the proteins appropriately, so they either get removed from the system or refolded appropriately. So that's one of their primary roles. And that's why they're induced during a stress response, so an ER stress response or an oxidative stress response in the cell. These induce these chaperone proteins, heat shock proteins, to respond, to then try and cope with the proteins, either going down a removal mechanism or staying fully folded and processing appropriately. Okay, so beta, it's a very important protein for controlling that. And just as an aside, my colleague works on alpha-B-crystalline because it's actually the major protein in your lens, in your eye lens, and actually is the main cause of cataracts. So if it aggregates in your lens, it actually causes cataracts, which I've got in my right eye at the moment, actually, just to bring it sort of. The age of 42 is the key age. So alpha-B-crystalline is very in the eye lens, and it's one of the primary proteins. In fact, it makes up about 80% of your protein in your lens. Anyway, but you get it in the brain as well.
So it's got to be very photoactive then. I mean, it has to have something to do with light. I mean, it has to be
potentially can be. Yeah, that's part of the mechanistic side of aggregation of the alpha-B-crystalline. Yeah, it could be a deleterious effect of UV light and etc. Yeah, so that's just as an aside. So again, this slide is really summarizing some of the key effects in the AD mice, so in the Alzheimer's disease mice. So you get upregulation again of these proteins I've already mentioned that we see in the CD1 mice, heat shock protein 105, 70, and the phosphorylated form of 27. So these are all three upregulated. Now you notice that the one panel on the right is the alpha-B-crystalline. So in the Alzheimer mice, what we see is, compared to a normal mouse, the alpha-B-crystalline is significantly increased, which is what signal you see on the left side. And then obviously with infrared light, you get a very significant reduction of that alpha-B-crystalline down to a more natural level, if you like. So it's a heat shock protein similar, or a chaperone protein similar to the others, but it has a different role in this story. Okay, so again, the infrared light of this particular wavelength seems to have these positive effects on the three heat shock proteins, but it has this inhibitory effect on alpha-B-crystalline expression. Nice. Okay, and again, just below each one of them, I've listed some of the roles of these proteins. So as I said, the heat shock protein 105 is a member of the 70 family, so it's able to substitute for heat shock protein 70 as well in some situations. So you can almost put those two together, and again, the light seems to induce gene expression and protein expression of these chaperone proteins. And it's induced, during cellular stress, and it prevents aggregation of damaged proteins, as I alluded to. So it sort of ties in with what I'm going to show you in a minute in terms of the amyloid effects. And similarly, the alpha-B-crystalline is involved in the aggregation and worsening aggregation process. Right, okay, so in terms of some of the key proteins related to Alzheimer's disease specifically, so in these AD mice, again, remember that they're 12-month-old AD mice, females that have a serious sort of memory deficits at that time point. And in this case, again, the light has some beneficial effects. It seems to reduce this phosphorylated form of tau. I'm not shown on this slide, but it doesn't seem to affect the overall tau protein. So this is a microtubule associated protein that's hyperphosphorylated in Alzheimer's disease, and is believed to be one of the primary molecular changes you see. It's more important probably than amyloid.
The microtubule is probably more important than the amyloid or the tau. Which one is it?
Well, again, it's been a controversy for the last 30 odd years, which comes first, which is the most important. I think the take-home message is, they're both important, but I think the take-home message is that the phospho-tau is probably more important to the pathology. And the main reason for that is that there's no correlation between amyloid load and learning deficits in Alzheimer's, whereas there is a clear correlation between the phosphorylated tau levels and memory processing.
So tell me, just because everybody, we've been the buzzword a little bit in the conference, the virtual summit so far has been the microtubules. So tell me, and in the microtubules, sort of the proteins on the microtubules sort of decouple and they recouple and things like that. So
Yeah.
you have a little picture here. So you said the microtubule is actually disintegrating. Well, so where does the tau clump attach if it doesn't have a microtubule?
Okay. So microtubules, so the way to look at it, if you think of a structure of a neuron, you've got a cell body and you've got a process. So the microtubules really run along the along the processes, and they transport, all the key elements up and down. It's like a train track, running down, up and down to the synapses, to the terminals. And they do, yes, they are made and lost continuously as neurites progress and grow, and then over time can potentially be lost and then regrown and things like that. So the microtubules are fundamental in the structure and the actual transport of organelles of various useful things. Now, the way the microtubules are structured, you need microtubule associated proteins attached. They're part of the track, so it's almost like the bolts that tie the microtubules together. So if you and what happens in Alzheimer's disease, for example, you get, in normal situation, you get phosphorylation and regulation of those processes, but in Alzheimer's you get hyperphosphorylation, so you get highly charged tau proteins. Okay, when you've got highly charged proteins, it's almost like a magnet. They basically affect the structure, and they disrupt the structure of the microtubules which are underlying the tau proteins. So you start to disintegrate the neurites, and so you lose the processes, you lose the terminal connections, etc., etc. So it's the hyperphosphorylated state is the key thing of those bolts, if you like, for the train track.
Say like rocks falling on the track until it's disintegrated, so the stuff can't get to where it needs to go, and the cell's not going to function. Is that how it works?
Potentially, but it's more. I would look at it more as the bolts that hold the rail tracks together. So they're needed for the actual structure of the microtubules. The microtubules are the tracks, and you need the bolts to tie them, hold them together. And if you get those disrupted, they will then, if the bolts become loose, your track becomes disoriented, and then you lose the structure.
So then the phosphorylated tau comes in and it helps disintegrate help take the bolts apart, or is it clogging up the railroad?
Well, it does both pretty much. It clogs up, but it also it disintegrates the structure of the actual microtubules themselves. So it's because they're highly charged. You think of a phosphate group on a protein. If you have more than one, more than a few phosphate groups, you become more and more negatively charged, and then you've got negative against negative, and it's repulsion of structures. So that's basically how it works. So it's misprocessing of proteins. Mis-signaling, modification of proteins. That's the problem. And it seems, again, that those sort of processes are controlled by chaperone proteins. Again, if they're not produced and structurally made appropriately, and you start to lose structure, then it falls apart. So if you, in this case, you're modifying you're increasing the chaperone proteins, you're increasing the chances of actually repairing the situation and getting appropriate phosphorylation, appropriate processing of the protein, and therefore you lose, that the light seems to reduce the number of hyperphosphorylated tau proteins, and therefore you got more chance of stabilizing the microtubules as a consequence. That's the simplest way of putting it, I guess.
Very well. Hey, Joe the plumber can understand it. That's good.
Okay, so that's the tau protein, which is obviously a good effect, again, useful effect. Now we get to the amyloid. So within this mouse model, you get amyloid deposition. And again, this slide is just showing you some of the structures that I'm focusing on. The cortex. These are part of the hippocampus, CA1, 2, 3, dentate gyrus, which I've already shown you before. So these are plaques, the amyloid plaques labeled by antibodies, specific for the A-beta proteins. And you can see large plaques, and you can see medium-sized plaques, and you get little plaques as well. Potentially more oligomeric type structures. And you see with the infrared light, even by eye, you can see the background amyloid, which are the sort of more soluble, which are the more probably toxic amyloid, is significantly reduced. But as well as that, you start to see less plaques as well, particularly when we measured them, the smaller plaques, the ones that are more closer to the oligomers rather than the larger plaques. So that's just pictorially showing the effects. And again, by eye, you can see the effects in the hippocampus and cortex, which are two key areas that control the synaptic plasticity and control learning and memory processing. So it all ties together, if you like, with the memory changes. Again, it's a bit easier to see. Again, this is an age dependent. So this is in, I think we're in the cortex here. Again, at 3 months, you can see a few plaques, and then 7 months and 12 months, you can see progressive increase in plaques of different sizes. In the cerebellum, which is less affected in Alzheimer's disease, you can see there's less obvious plaques in there. Then we close in on one area here. You've got lots of plaques here, different sizes. And again, with the treatment, again, this is 3-month treatment, progressive treatment over 3 months. You see again, you see a reduction in signal, general signal. So that presumably is the oligomeric smaller stuff within cells, less, as well as the plaques are reduced. And this is the most stark result we ever got, actually. I couldn't believe it when I saw this. That's the protein when you label it, and then with the infrared light, it's clear, massive reduction, which reflects, the change in amyloid load with this treatment. And again, the assumption is that's to do with the changes in chaperone proteins and how the protein's processed is resulting in this massive change in amyloid load. So it's been cleared much more easily from the system because you've got increased chaperone proteins to do it. All right, that's the idea, basically.
Gotcha.
All right, so, but as well as that, we did try and look at the plaque sizes and things, and we did notice, that's not as dramatic as the overall change, if you see here, but even with the plaques, with the smaller plaques, you see a significant change in numbers of these smaller plaques, whereas the larger ones were not so obvious. Again, it all correlates with this reducing the more important plaque, more important amyloid, i.e. the smaller stuff and the soluble stuff is being reduced by the treatment. Again, that's just an example of the data. Okay, so this sort of summarizes some of the things. It's not, hopefully you can see this. So again, heat shock proteins are in this diagram in amongst looking at the mitochondrial effects and looking at the processing of the amyloid effects. So this is heat shock protein 70, which I've highlighted, because we'll talk about it in a subsequent slide. So this is one of the proteins we've been focusing a lot on, is heat shock protein 70, because that's involved in the aggregation and the changes in amyloid load that we talked about, and the changes in mitochondrial function also, state 4 in particular. So we're going to talk about heat shock protein 70 in more detail just for the last couple of slides. So heat shock protein 70, just again, I've focused on this. So this is very much involved in looking at changes in affecting misfolded proteins and also the effects on mitochondria. So these are the two effects we see with the light treatment, the two main effects we see. And heat shock protein is upregulated significantly both in the aging CD1 mouse as well as the Alzheimer mouse, and it correlates with the amyloid changes, etc., and the phospho-tau. And that's the protein we're now focusing. So we did then an experiment, again unpublished, where we've looked at the effects of the light therapy on a simple model. So the C. elegans model is a very useful model for studying aging.
What is C? What is that? A worm? Is that what is that?
It's a C. elegans. Yeah, that's the worm. So it is a type of, it's a type of roundworm. So it's a very small worm. It's only like a few millimeters. This is extremely magnified. So this is it's a very commonly used model system to study aging, because these things only last for a matter of a couple of weeks, and you can study them in great detail. They're particularly useful for some of my colleagues use it looking at diet and the interaction with bacteria. So you can use it as a gut model system to look at gut aging and things like that. But obviously it's fairly rudimentary in terms of the nervous system. But in terms of aging, what we did, we treated, again, multiple treatments without, one of the key things we developed in this experiment is to make sure that we didn't get a change in temperature, because the device that we use in all our studies can potentially generate, just technically can generate a temperature change. That was not an issue when you're dealing with mice and bigger things. But when you're using the worm, you have to be very careful you don't get a change in temperature when you're treating them. So we had to adapt the methodology to do that, to make sure we developed a method to actually measure the temperature in this system, which was not been published before. And we showed, we used a treatment system that didn't change the temperature, significantly. Because it's quite important, because if you change the temperature within these, you can actually affect aging lifespan as well. It's a well-known phenomena. So that was a very important thing to do. So within this experiment, we changed the protocol. So we should use shorter exposures to the light over longer periods. So we didn't change the temperature. And using that methodology, we showed an extension of lifespan. It's not particularly dramatic, but it's significant. A small change, which relates to about 16% change in lifespan. So numbers, more numbers survived. So that doesn't, if you're comparing that to other methods of extending lifespan in this system, you can get double lifespan times for a lot of other treatments. So it's quite a modest effect, but a significant effect. But the key thing with this experiment was we then tried it with a mutant worm that didn't have heat shock protein 70, and you lose that effect, basically. Losing heat shock protein also affected lifespan on its own. As you can see, the control is much quicker here, but the effect of the light was lost, showing that this effect was partly to do with heat shock protein 70.
Very good.
So that was the idea of the experiment, and that's the beauty of using this type of model system, because you can look at particular genes that you're interested in to study the effects of your treatment to give it an idea again of mechanism.
Cool.
So this is some evidence for heat shock protein 70 being quite an important part of the story. Okay, so that's, we like I said, we do start to work on more relevant models now. So we moved on from C. elegans to flies, because again, you can do lifespan experiments with flies, and you can do behavior experiments, and it has got a rudimentary nervous system. So it has many of the neuronal systems that you see in a mammalian brain. A few less of them, obviously, numbers, but the actual system is pretty much the same. So you can actually study motor effects. You can look at memory performance, there's, which we're using at the moment, and you can also even look at social interactions within these flies. So we've developed a method of doing that. So we've got sort of an all-in-one behavioral screen for flies that's analogous to our mouse system. And with that, you can start to treat the flies with light. And this is just an example. This is looking at motor. So this is a sort of a PINK1 mutation, which is a mitochondrial protein mutation in Parkinson's. And you see motor deficits. In the healthy wild type, the flies are very mobile. And with the light treatment of that mutant, you can start to see improvements in motor function. This is early days, pilot days. So we're working on that, but it seems to be a model.
Powerful observations.
Potentially, yeah. There are caveats, because the fly brain, even though I say it's got a rudimentary nervous system, it does lack some of the targets, some of the protein targets that you see in a mammalian system. For example, one of our other projects, we're working with retinoids as retinoid therapy for ALS at the moment, and the target for that particular retinoid is not found in the fly, so we can't do those experiments in the fly. It's lacking the actual target, so it does have some
the picking of the species allows you rapid results. You'll be able to see the stuff quickly. So I think as a preclinical model, you're right on it.
Yeah. So it's for example, you can use it to look at the innate immune system. It's got actually quite a nice model for the innate immune system. You can study Parkinson's because you can measure dopamine. It's sensitive to levodopa treatment, for example. So it's like a mouse model. It behaves exactly the same way. So there are dopamine neurons that you can lesion, and you can study, and you can do biochemistry. You can do oxidative stress markers. You can do ER stress markers. You can look at all the various changes you see in an Alzheimer's brain in mammals. So it does have some strong potential to study. I know there is some. I mean, there's been some papers with light therapy in flies, and there's been sort of a mixed opinion about the use of the fly model. Some people have found it useful, some less so. So it's still a work in progress, I think, as a model system. But we started to use it, and we found some uses for it so far. But within the restraints I've alluded to. Okay, so just to summarize, really, we're getting to the end now. So just summarize some of the effects that we're seeing with our particular wavelength. And again, some of these are common to other wavelengths, like the 617 nanometer wavelength. Some of these effects, like increased ATP production, for example, and which I didn't really touch on, but we see that, and we see changes in particularly state 3 and 4 for mitochondrial function. 670 seems to modify maybe complex 2 and 1 more readily as well. Increase in nitric oxide, as an example, a mild increase in reactive oxygen species, which goes together with the mild stress idea. So this being a preconditioning mild stress inducer, which then results in gene expression changes, which then results in upregulation of chaperone proteins in response to the stress, and other signaling molecules, which ultimately leads to changes in things like amyloid and tau, phospho-tau, etc. Okay, so that sort of basically summarizes, and I'm just going to quickly go through some human studies that have obviously come out of our work. This is from Marvin Berman's work, who's now running these clinical trials, which I guess some of the guys watching this who will know him. He's presented some of this stuff, I'm sure, in other meetings. So I'm just going to rattle through some of the things. So, we're starting to use them in human models. These are independent studies being carried out in the States. And this is just an example of some anecdotal or some pilot studies that have been done with this particular wavelength. And I know, other studies have been done with other wavelengths, particularly 670 and 850, following similar protocols, similar paradigms. So, we get, clear effects on within Alzheimer's disease cases, and there's also been some small studies in Parkinson's cases as well. So this is just some examples of, before treatment of a poor performance in terms of drawing the clock as a sort of classic test for cognitive processing and being able to, to draw it accurately, draw a clock accurately. And obviously after 5 weeks treatment, again, multiple treatments, that's the key, multiple treatments with the light, you get an improvement in that sort of performance and that sort of task. And again, you get physiological changes, which again, it may not be too clear on this slide, but you get changes in different power settings within the brain, so different electrophysiological changes, if you want to put it a better way, which allude to improved alertness, less anxiety, and increased focus and memory performance, as suggested with and without the treatment. So there's physiological changes we're seeing in human cases based on the treatment, which go along with the idea of some of the behavioural changes we see preclinically. It's early days, but it seems very, very promising as a wavelength potentially to change some of these useful properties. Again, improve changes in delta waves, which can be correlated to mood and sleep performances and neural processing, for example, here. So again, I'm rattling through that quite quickly. Obviously you can look at that. I mean, Marvin is probably the best person, again, to talk you through that. If anybody in the audience is interested to discuss that further, this is a bit out of my remit, if you like. But it's showing some promise, early promise. And again, this is data that was published recently with this wavelength. It's in the first study again, a pilot study with a small number of individuals showing, again, some improvement in, particularly in things like delayed will recall, so cognitive memory performance, attention performances seem to improve with the treatment. Small numbers, but pointing, in the right direction positively. Importantly, there's no indication of negatives, which is equally important, to be honest, in these studies. And also we've done some recent stuff. Again, this is unpublished, showing some effects in actually healthy, aged individuals rather than disease cases. So we've done this actually in the UK and in Europe. And again, it's pilot studies, it's being basically put together for publication. But just to summarize some of the positive effects, again, it's a small number, relatively small number we're looking at. But there's things like mathematical processing, i.e. working memory, processing speed and delay memory tasks, and even some motor benefits, it seems, from the treatment. Small numbers, but again, all pointing in the right sort of direction and correlating with some of our preclinical. It obviously needs bigger numbers, and there's an ongoing recruitment for a larger trial in Alzheimer patients at the moment in the States, and we are developing a bigger study with the aging population in the UK, because we can't get this done in the UK, anything to do with disease cases, which is a big problem in the UK.
Well, that's why you make lemonade when they give you lemons. You just need a little sugar, But if you can't do the clinical trials, you do the preclinical and excel at.
Yeah, exactly. Yeah, I mean, it is one of the biggest problems. If you're talking about issues about this sort of work, it's actually we've had great difficulties being able to get approval to do trials with this type of treatment, any sort of wavelength, in the UK for disease cases. Unfortunately, we've tried many times, and it's the ethical aspects. I'm still trying to get my head around it. In fact, it's a general problem. It's not just about light. Any trial in the UK is so much more difficult to do.
When you think about this, 35 patients in this early 35 participants, that's a large number for a transcranial study.
It is, absolutely.
And the numbers, the changes that you're showing now, we don't have all the details of what the outcomes were, but it's moving the needle. We're seeing that it's making changes just by looking at the data that you put.
Yeah, absolutely. Yeah.
Quite positive that you're
absolutely
being
it's going in the right direction. I mean, there's no for me, it's more important to see there's no negative. I mean, I have to be really honest, actually, in all our preclinical work, I've looked for it, but I've not actually found anything particularly negative in terms of its treatment, which is equally important, actually.
And this is, the research has always shown that, and that's what we've seen. So, I mean, from the microscope to the telescope to the future scope, Paul.
Yeah.
We're at the 2020 Brain and Transcranial Photobiomodulation Virtual Summit. Let's tell me what's going to happen at the 2030. What are we going to see? What are we going to see with this science and technology? What do you see happening in the future of this process?
Well, I'm pretty optimistic, to be honest. I'm sort of generally optimistic anyway, but about many of the things that we're doing, because we understand the biology a lot more now. We're underlying the system, and we're starting to really tease out the mechanistic side of what's going on here. We're not there yet fully, to be honest, but we are some way down the line, and there is a really impressive growing evidence base that underlies the rationale for doing it. Again, we need to probably think about optimizing the system, because that's one of the main problems for us. Is we've sort of made guesstimates of the sort of protocols and the treatment. We've done our best. We're limited by funding. We're limited by being able to do that, to optimize the system. So we need to do a bit more on that to be able to optimize the system to get maximum beneficial effect with obviously no or little side effects. So that's a work in progress. But I see 10 years down the line, yeah, I mean, the way the early data is coming out is not just from our wavelength, you know, the 670 is starting to build significant evidence base clinically that we're going to, you know, we're going to have some useful effects here.
Again, so we're going to see, you know, a lot of the bench work that you've done is going to allow us to tick-tock and, like you say, I like the word tease out, but I actually, you know, have a more appropriate targeted response,
yeah,
and allow the indirect, you know, the direct response is going to be a little more targeted, but our indirect response is just going to come along for the ride, right?
Yeah, sure. Yeah, yeah. But I am, yeah, like I said, I'm very optimistic. I mean, I think, you know, we are some way down the line now, and there's been some great work going on, particularly in the States, actually, you know, in this area that's really helped us sort of start to change the minds of skeptics. I mean, like I said, at the beginning, I was a skeptic, you know, right from the beginning. When Gordon approached me, I thought, okay, we'll give it a go. He's an interesting, eccentric South African, you know, I'll go with it, see how we go. And, you know, that from that first experiment, I was hooked pretty much because it, you know, again, I was surprised by the result, you know, that it was doing something. Then I started thinking about it. And once you start thinking about it, you start to, okay, that makes sense. That makes sense, you know. And the evidence base is building, you know, there's a lot of great stuff going on in the States, like I said, that's building on that evidence base. So I'm pretty optimistic that we're going to get there. But I think, like I said, we need to consider, optimization of the system, and we need to maybe make a decision again of wavelengths, and maybe we might need to consider combination of wavelengths. Maybe that might be add to the mix.
Investigate them all. You know, like I said, you have got a nice model, very quick and fast, and we can get the results. So you can do it. You know, you don't have to do it in the cell culture at this point. You can do it in the fly, and boom, boom, 2 weeks, you're done, and you run the next frequency and the next wavelength, and away you go. And so, you know, I think that's a great model, and that's what we need to have done.
Sure. But I mean, there is, like I said, there is some caveats there. We have to be careful, you know, always along the way, because, like I said, the fly doesn't have everything. You know, maybe some of the targets that are missing, like I said, with our retinoid approach, you know, that again is the same idea. It's actually using a retinoid to target multiple mechanisms, genomic and non-genomic, to give you, you know, multiple effects within your disease. But yeah, you have to have the targets in the system to be able to take them to.
As a takeaway, you know, I'm sure that the flies are very happy that they got 16% more life. You know, when you look at the human, that could be an extra dozen years. So
yeah, absolutely.
A good life, not a diseased brain life, but a healthy memory functioning life. So that's
the potential. That's the potential. Yeah. But as I said, that wasn't optimized. So, you know, we could have got better. I mean, we tried a protocol based on our cell work, but, you know, it may not have been the optimal. So, you know, there's the potentially that's probably the minimum we could get. So there's always that to think about as well.
Have at it. Thank you for your time. Thank you for your expertise. Thank you for all the work that you've done.
Okay. Thanks a lot.